Now we turn to Dr. for Clinical Methodology at CDER of the US Food and Drug Administration. And Leonard, the question for you is, how can stringent regulatory agencies such as the US FDA be innovative in facilitating the regulatory environment for the drugs for neglected diseases?
Well, thank you very much, Nick, and thanks to the organisers for having me here. I spent a little bit of a sleepless night pondering that question, and my eventual conclusion was that we probably do have some role, and I was going to tell the story as follows. I think the history of this really does go back to 1981 approximately, and we've heard this before, when there was the sudden emergence of a terrible disease, a disease with no treatment, a disease which people were dying from, and a disease which basically affected a marginalized minority where the industry was not very excited in terms of developing drugs. But thanks to a lot of advocacy and sort of lobbying at that time, there was an enormous groundswell in support for regulatory changes. And the things that were introduced at that point were a couple of incentive programs, regulatory incentives. These included things like what we call priority review and accelerated approval. These are programs that shorten the duration of review and that basically allow the approval of drugs on the basis of a surrogate endpoint, provided that can be subsequently clinically substantiated. So these were two important mechanisms. The third thing that I think played a crucial role in the success in the HIV environment was the development of a surrogate endpoint. And I think these are three sort of areas which have some reference to neglected diseases as they stand now. Just to remind ourselves, the ability to use the viral load as a surrogate endpoint for clinical outcome in AIDS was a tremendous breakthrough, and it basically converted the follow-up time in a clinical trial from effectively years to a period of 6 weeks. So looking at those efficiencies, I believe that some of them have been applied to neglected diseases. Certainly in my tenure at FDA over the last 15 years, I've seen a couple of diseases moving from neglected to not neglected, TB being one, malaria being another. And we do have a couple of new products for those that have been approved. So just to summarize some of the areas where I think regulatory agencies can provide some help in getting efficiency improved. The first one, as I mentioned, was these incentive programs, and we do have a number of them. I mentioned priority review, which shortens the time of review within the regulatory agency. I mentioned accelerated approval, which allows for the use of surrogate endpoints. There are things like orphan drug designation, where if the disease occurs in a minimum or at least a limited number of patients in the United States, user fees are waived. There's some incentives given to companies in terms of prolonged exclusivity and so on. We have the priority review voucher system, which rewards organizations that are able to develop drugs for these diseases of need. And the very latest designation, which some of you are probably familiar with, is the breakthrough designation. And I think this is another attempt for FDA to try and encourage very early consultation with FDA when a new product looks as if it's going to change the paradigm and where there is at least a little bit of supportive clinical data. So I think these are very helpful incentive programs. There are a couple of other areas that I thought I should just mention. I'm sure many of you are aware of them. There are other pathways for the submission of applications to FDA. We have a 505(b)(2) pathway, which effectively allows the use of referenced literature to obtain licensing. And although this wouldn't apply to most of these new products, I can certainly think of it as being useful, for example, in things like nifurtimox and benzonidazole, which have never been approved in the US, where literature-based submissions would suffice or may suffice to get regulatory approval. The other one, just in passing, is the animal rule. sounds a little barbaric, but really that's just basically a mechanism to approve drugs based on animal data alone, and that only is applicable when there is no human model. But I think it's worth bearing that in mind as a pathway for diseases like rabies, for diseases like Ebola, for diseases like Marburg. So there are a couple of other pathways. I wanted to move, or at least shift gear slightly, and touch on the issue of foreign studies. I think a lot of people shy away from the idea of completely foreign data to service FDA applications. In fact, it's interesting that about 60% of all the pivotal studies submitted to FDA are conducted overseas, or at least partially. So we're very familiar with foreign data, and we're very welcoming of foreign data. And provided we can ensure some basic standards of good clinical practice and so on, that's a very fundamental source of global drug development. I wanted to turn to another opportunity, which is something that certainly got my attention, and this is the sort of new universe of technology, which I think we've underexploited in drug development. You know, we've had a lot of success in sending rockets to Mars, and it seems a little bit surprising that we haven't employed that degree of technology in clinical drug development. The point being that we're trying to move clinical development into a completely electronic platform so that clinical trials are not conducted on paper. They can be conducted in a centrally based electronic system. The data can be rapidly transmitted. The data has a lot of quality controls in terms of date timestamps, audit controls, and so on. So these technological electronic platforms, to me, are a great opportunity, and they serve as the basis for a couple of other things. First of all, there are many things that can plug into these case report forms. Informed consent can be an electronic added point. Mobile applications can be an added input. For example, patients may be able to communicate via cell phones and so on, patient-reported outcomes. There may be physical transducers that can send signals into these case report forms. For example, a mobility transducer in patients with a movement disorder, or a seizure monitor in patients with a seizure disorder. So there are many technological opportunities that I think we have to consider as new ways where regulatory organizations can advance the cause. And the last strategy, which I just wanted to mention, and I hope you will bear with me, was something that I've shared with a couple of members of the audience. I know that this was raised several times during the morning, but repurposing seems to me a very profitable strategy from a regulatory perspective. First of all, drugs which have been used for other diseases are already characterized in terms of their safety, their pharmacokinetics, and their manufacturing. And we know from our previous experience in neglected tropical diseases that repurposing has been one of the backbones of servicing these different diseases. We know, for example, that amphotericin, that the azoles have very promiscuous sorts of activity in these infectious diseases. And it's also worth bearing in mind, for example, that things like miltefosine, like eflornithine, like AZT were all cancer drugs that were repurposed. So we think this is a fruitful area, and actually one of the projects that I'm sort of encouraging at the moment is the development of an internet-based data system where if physicians out in various parts of the world, in Burundi, in Burkina Faso, in Fiji, in Brazil, have experience using drugs for different diseases, using novel combinations, using new doses or new durations of therapy, we will be able to provide an internet-based questionnaire, a very simple one-page questionnaire, to gather this information from providers in the field. And that way we will have some method for integrating areas where drug-disease combinations look promising. It certainly was my experience early in the TB world that there were several missed opportunities in this respect. For example, I think when drug-resistant tuberculosis first emerged, there was a lot of panic among the medical practice community, and everything was tried. People tried Augmentin, they tried imipenem, they tried quinolones, and they tried linezolid. And the latter two turned out to be very important treatment options in tuberculosis, but really this came to the fore very late. And I think with current electronic opportunities, that sort of information can be gathered very early. So it's just a plea that at some point we're hoping to put out such a program. I think it won't be too long in the future. And this will rely on global cooperation to get the data from all over the world. But, so those are some of the thoughts that I had, and I think that's as much as I have to say.
So we've seen the US FDA becoming much more accommodating. What would you like to see from us on the other side who are developing these drugs? I mean, you've made a strong plea for electronic data records. We've heard that. Are there other things either in terms of practice or attitude that you think would facilitate and accelerate development of drugs for neglected diseases?
Well, first of all, I have to thank you for the compliment. Second of all, I think that the difficulty we face, obviously, is that FDA is basically committed to a uniform standard that drugs are meant to be safe and effective. And really, the only sort of alterations in that equation are determined by the risk benefits. So if you're looking at a very high-risk disease, obviously one's prepared to trade off a certain amount of safety data. But I think that we definitely want to try and maintain that quality. And I think part of the way this could be done would be by education, by spreading expertise, by building clinical study networks which support the acquisition of high-quality data. And then, of course, supporting electronic infrastructures may be another way of bringing this together. Just another point was that biomarkers are probably a very important tool that we may not have emphasized sufficiently in this meeting. But I mean, when thinking about chronic diseases like Chagas disease, like lymphatic filariasis, we really don't have the tools to develop drugs for these conditions in short periods of time. And I think there the scientific and the drug development community can help us a lot in looking for biomarkers of efficacy.
Can I just take up on that particular point? Because it seems to me that what you're saying is we haven't spent enough effort in characterizing pharmacodynamic endpoints and therefore characterizing pharmacokinetic, pharmacodynamic relationships. And the consequence of that is delayed, very slow drug development, more costly drug development, and often the wrong dose. Would that be fair?
Oh, absolutely, undoubtedly, Nick. I think that's something I mentioned it a little earlier privately, but we did a recent review of drugs which failed to get approved at FDA over the last 12 years, and it was very clear that dose or the incorrect dose selection was one of the most common reasons that drugs do not get approved, at least when they're first submitted to us. So I think that developing the pharmacogenetics is very important. I think thought should sometimes be given to late-stage investigation of dose ranging. I know that it's very seldom done in traditional development plans, but sometimes it's better to think about various doses in phase 3 trials than rather to choose one sort of one single dose to invest everything in one dose. And that can be done by not so much in infectious diseases and other diseases, I suppose. You can basically randomize patients to receive successive periods of different dosing and see whether you get different effects at the different doses. So I think some thought should be given to late-stage qualification of doses, but dose is a big problem.
Please come to the microphones if you have any questions. So on that particular last point, you would be happy to entertain a dialogue with a developer over the design of the phase 3?
Oh, undoubtedly. I think, in fact, FDA has been very enthusiastic to do that, and I think the literature encourages that in terms of product success. Yeah.
Thank you. So, yes, we have a question.
Could you say a few words about the current status of the FDA Orphan Drug Program and its relevance for this particular community?
Sure. So the Orphan Drug Program is really designed to promote the development of drugs where there's really very limited economic appeal. The requirements are pretty simple. If the disease is present in less than 200,000 individuals in the United States, the drug will qualify for, or generally will qualify for, orphan designation. What are the advantages of orphan designation? Well, the main ones are really post hoc. With an orphan designation, there's an added period of exclusivity where no generic competitor can subsume the market. There's a waiver of the user fee, which can be quite pricey for small organizations. There's the opportunity to discuss these things in a little bit more detail with FDA representatives. So I think those are the main advantages of orphan drug designation. And I do have to say that of the so-called neglected diseases for which drugs were approved recently, all of them had orphan designation. Bedaquiline recently for TB, Coartem for malaria in the US, Malarone way back for malaria. They all had orphan designations.
Does it work? Yeah. I'm Patrick Dorisch from the Berne Declaration. It's a Swiss NGO advocating for access to medicines and ethical clinical trials. You mentioned this number, which corresponds to the figures I had in mind, of 60% of the data coming from abroad. I was just wondering, because we just made an investigation in 4 different countries, emerging countries, about the offshoring of clinical trials, more industry-sponsored clinical trials, not so much neglected tropical disease trials. And we found out that there were quite a substantial number of ethical violations due to the fact, I mean, there are several reasons, but one of the reasons is that the systems in place, the ethical supervision systems are not yet in place or are not working so much. I'm happy to hear that in Africa there seems to be kind of others. We haven't investigated Africa, but there seems to be quite a sort of community control about that. I was wondering, from the FDA point of view, how you check a posteriori the ethical compliance of those data, how they've been generated, and if the trials have been conducted according to the ICH-GCP or Helsinki Declaration standards.
Yeah, I think it's a very important point, and we inspect them. The overseas sites are inspected. I guess FDA tries to focus on areas where the risk is highest. Source data is evaluated, and post hoc comparisons are made between different sites, between US and between non-US. And on occasions, I have to tell you that there have been disparities. And at times, that has been an obstacle to drug approval. When drugs perform outside of the country, outside of the US, and they don't perform in the US, this raises serious concerns among reviewers. So I think it is very important to ensure the quality of the data that comes from overseas. As I say, inspections are one tool, but I imagine there are ways to do it. There are ways, at least, to improve that. One is educational outreach. I think at least when we have the opportunity to educate people outside in the performance of clinical trials, when external regulatory authorities are also empowered to do this, I think that will strengthen the quality of data. And finally, I still think that this is an electronic opportunity. I think when we can move to electronic platforms where the data can be queried centrally, where the data can be queried contemporaneously, so that we don't have to go back 2 years later and ask an investigator what happened to a patient that they've already forgotten about. When we use those electronic tools, I think we will be able to ensure that clinical trials conducted all over the world are conducted at a higher standard. So I think there are ways of tackling this.
Thanks very much, Leonard.